Every time you remember a face, solve a puzzle, or simply take a breath, it starts as a single microscopic spark. That isolated electrical impulse leaps across a synapse, triggering a cascading storm of activity. Zooming out from that single connection reveals the raw hardware of the human brain. It operates on a network of approximately 86 billion neurons. Supporting that network is a roughly equal number of glial cells, specialized cells that provide structural support, insulation, and nutrients to the neurons themselves. This animated diagram illustrates a stark biological imbalance. The physical mass of the brain accounts for only about 2% of your total body weight, yet it consumes 20% of your body's total energy, relying heavily on blood glucose. Millions of species on Earth have brains with a similar cellular makeup, yet humans are the only ones building skyscrapers, writing symphonies, and mapping the stars. To understand how we developed the capacity for abstract thought and complex civilization, we have to look past the sheer number of cells. A common assumption is that a larger brain automatically yields higher intelligence. But if we measure by raw anatomical volume, humans fall far behind species like sperm whales and elephants, which possess brains weighing several times more than ours. This chart compares the brain-to-body mass ratios across different species. You might expect humans to top the list, but the highest quotient actually belongs to the tree shrew, a small mammal native to Southeast Asia. If raw physical size and mass ratios fail to explain our cognitive abilities, the answer isn't found in how much space the organ takes up. The secret lies in the architecture of its wiring. The hominid brain underwent a rigorous period of physical expansion. This trajectory begins around 4 million years ago with Australopithecus, one of our earliest bipedal ancestors. Over millions of years, the cranial vault expanded rapidly. Early hominid brain size measured around 600 cubic centimeters. By the time Neanderthals and modern humans arrived, that average had swelled to over 1,500 cubic centimeters. But the expanding skull was essentially just building a larger chassis. The actual biological upgrade was taking place on the brain's outer surface, the cerebral cortex, a deeply folded layer of gray matter that crumpled and wrinkled to pack more surface area into the confined space of the skull. The neocortex is composed of 6 distinct neuronal layers, stacked tightly together. This specific microanatomy creates an incredibly dense web of synaptic connections. The vertical and horizontal circuits formed here multiply the brain's processing pathways exponentially. The evolutionary shift in human capability was driven by the density of the processing network rather than the sheer volume of the skull. Within that network, the highest level of processing occurs at the very front of the brain. This is the prefrontal cortex, the most highly evolved region of the human command center. This region drives what neuroscientists call executive function. It allows us to filter out irrelevant information, suppress immediate impulses, and manipulate working memory, the mental scratch pad we use to hold and process information in real time. Most animal cognition is tightly bound to immediate sensory input and reflexive action. Humans use the prefrontal hub to detach from the present moment. We mentally simulate different scenarios, weigh long-term consequences, and plan for events that haven't happened yet. By developing this biological engine, early humans severed their strict reliance on instinct. We gained the power of abstract foresight. Abstract thoughts are useless if they remain trapped inside a single mind. To share these internal simulations, the prefrontal cortex requires dedicated neural highways to format and transmit information outward. This left hemisphere map identifies Wernicke's area for language comprehension and Broca's area for speech production. Bridging them is a bundle of nerve fibers called the arcuate fasciculus, linking our ability to understand words with our ability to speak. By linking internal comprehension to external production, the brain enabled the transfer of complex knowledge between individuals. This ability to coordinate and pass information down through generations transformed a biological network into a social one, providing the foundation for shared culture and complex human societies. Today, we sequence genomes and send rovers to Mars, yet the exact operational parameters of our 86 billion neuron network remain one of the ultimate scientific mysteries. Historically, attempts to map the mind relied on guesswork. In the 19th century, the pseudoscience of phrenology localized personality traits by measuring bumps and drawing arbitrary boundary lines on the outside of the skull. We have since abandoned the skull to map the wiring itself. Launched in 2009, the Human Connectome Project is a massive, data-driven endeavor to definitively map the brain's complete anatomical and functional connections. Researchers now use advanced imaging techniques like tractography, using magnetic resonance imaging to trace water diffusion, creating highly detailed 3D models of actual nerve tracts. These maps serve as a diagnostic key. By tracing these actual nerve tracts, we can identify the mechanical failures behind mental disorders and track the physical progression of neurodegeneration. The human brain is a self-contained biological supercomputer. Through millions of years of rigorous physical expansion and internal rewiring, it evolved the precise processing power to finally study its own source code.